students of class 9th. In previous video, we had studied about importance of fungi up to in preparation of beverages. We had studied the importance of fungi, that you can say yeast, in the process of beverages, in the preparation of beverages. Now, second use we are going to study in baking, that is in the preparation of bread. In this process, the baker mix prepares the dough in the evening by mixing flour and water. Suppose this one is dough you know, atta, gutha hua atta, and in this dough, this is the container, in this dough, the baker will add yeast powder. Yeast powder purchased from market. This yeast powder contains this yeast cells. Yeast cells. These yeast cells which are added inside the dough will perform an aerobic respiration throughout night. This dough, after preparing dough, it is by adding after preparing dough and adding yeast powder, this dough is left for whole night. During this whole night, the yeast cells will respire anaerobically, and already you have studied about the anaerobic respiration of yeast. This one is the anaerobic respiration. The equation of anaerobic respiration, you see, in this anaerobic respiration, ethyl alcohol is produced. This ethyl alcohol is produced inside the dough, as well as carbon dioxide gas is produced. This carbon dioxide gas will occupy some place, since it is in gaseous state. So this gaseous state will occupy some space inside the dough, and due to occupying this space, it will rise the dough. It will rise the dough. Dough means halka ho jana, फूल जाना. So this dough will rise. This rising of dough by the anaerobic respiration of yeast cells is known as leavening. L-E-V-E-N-I-N is known as leavening. Clear? Now, in the morning, this dough is taken inside the molds, sancha. This dough in the morning by baker is taken inside the molds, and the molds are kept inside furnace. Inside furnace, the dough will be heated, and the carbon dioxide gas, which has occupied some space in the dough, that will escape out. Due to escaping out of carbon dioxide, the dough will become soft. That is, you can say bread will become soft. In this way, the baker prepares the bread inside bakeries. The baker prepares the bread inside bakeries. Clear? This is the use of fungus in first baking. Already we have studied about beaver. Now, third one is cheese. What is cheese? Cheese is a valuable food material. Cheese is a valuable food material with high quantity of fats, proteins, vitamins, calcium and phosphorus. Vitamin A and vitamin B are also found. So you can say cheese is a valuable food material which contains high quantity of fats, proteins, calcium and phosphorus, as well as vitamin A and B. This cheese is prepared from milk, and this preparation of cheese from milk involves 4 steps. What are those 4 steps? First one is curdling of milk. The milk is converted into curd. Milk is converted into curd by adding Lactobacillus. Curd is converting in milk is converted into curd by adding lactobacillus. This process of converting milk into curd is known as curdling of milk. The curd which is obtained, the curd which is produced is separated from whey. Whey, you can say, liquid part of the curd, that is known as matta. The curd which is produced is separated from the whey. Clear? This is the fourth system, curdling of milk, in which the milk is converted into curd by adding lactobacillus, and the curd which is obtained is separated from the whey. Second step is processing. In case of processing, the curd is processed to obtain cottage cheese by removing moisture from this curd. The moisture is removed, and after removing the moisture from the curd, the rest part is known as cottage cheese. This is the second step. Third is salting. Salting of this cottage cheese further removes the moisture. If any moisture is left, this leftover moisture is removed by the process of salting, and due to removing the moisture from the cottage cheese, this salting prevents the growth of harmful microorganisms. Next is ripening. In ripening, the curd is kept at a suitable temperature and humidity. In ripening, the curd which has been completely dried, from which moisture has been completely removed, is kept at suitable temperature and humidity to provide particular flavor by which is produced due to added lactobacillus. That is, this cheese from which the moisture has been completely removed is kept at suitable temperature and humidity to provide particular flavor by added lactobacillus. In this way, with the help of by the process of these 4 steps, we can prepare cheese. Clear? Now next is edible mushrooms. There are different types of mushrooms. For example, molds, and mushrooms like there is toadstools. There are some of the mushrooms which are highly nutritious, and due to their highly nutritious nature, the mushrooms are eaten by humans due to edible nature, due to having nutritious value. The mushrooms are eaten by the person. Now, there are different species of mushrooms. Now, the edible mushrooms, due to their nutritious value, the edible mushrooms are grown or cultivated by the human beings. There are 3 types of species which are cultivated by humans for edible purpose. What are those 3 species? First one is white button mushroom. It is known as Agaricus bisporus. White button mushroom is also known as, in biology, Agaricus bisporus. Second is paddy straw. This is known as wall variella. And third is oyster, known as pleuropus. In this way, these 3 types of mushrooms are cultivated by humans. Now, mushrooms, for cultivation of mushroom, that is also cultivation of mushroom also involves 5 steps. What are those 5 steps? First one is composting. Composting means that is mushrooms are grown on decayed organic materials, on decayed organic materials. This decayed organic material is acted upon by means of they are recycled. So you can see mushrooms are grown on decayed organic materials, and in case of composting, the compost is prepared first of all. Compost means that is decayed organic materials, and it preparation of this compost involves the mixing of, again listen, preparation of compost involves three main components. First one is wheat or paddy straw. इन हिंदी पुआल, धान का पुआल होता है, वहाँ पर देखते होंगे ये मशरूम्स जल्दी ग्रो कर जाते हैं. तो wheat or paddy straw. Second, chicken manure, and third, some organic and inorganic manures. These three are mixed in proper proportion to prepare compost. Clear? The first step is composting, preparation of compost. After mixing, the compost is kept at about 50 degrees centigrade. Is kept for about 50 degrees centigrade. For how many days? For near about 2 weeks. After preparation, the compost is kept at about 50 degrees centigrade for near about 2 weeks. Now, second is spawning. In case of spawning, mushroom seeds means mycelium. Already we have studied mycelium of selected mushroom, which species you want to develop. Mushroom seeds or mycelium of selected mushroom is introduced inside the compost. And it is allowed to spread for a couple of days. This one is compost. The mycelium of selected mushroom is introduced inside compost and is allowed here to grow for about it is allowed to grow for a couple of days. This one is known as spawning. Now third is casting. A thin layer of soil is spreaded over the compost, is spreaded over the composting. For what purpose? There are three important functions of this soil. Number one, it prevents the drying of the compost, that you can say it helps in providing humidity. Number two, prevention of dryness, and number three, regulation of temperature. This soil, and there are soil which is added, performs different functions. First, it provides moisture, it prevents the loss of excess water from the composting, and next is regulation of temperature. This spreading of thin layer of soil is known as casting. Clear? Now next is cropping and harvesting. Mycelium grows within 2 to 6 weeks. Mycelium which has been added here will grow on the entire composting. Within 2 to 6 weeks, they form pin heads. And these pin heads are allowed to develop in the form of button-like structure, and these button-like structures are again allowed to grow up to marketable size, so that they can be brought inside the market. This one is known as cropping and harvesting. After that, the main important function is preservation. Mushrooms are highly perishable. बहुत जल्दी नुकसान खराब हो जाते हैं. So to provide, to increase their shelf life, mushrooms are highly perishable. So to increase their shelf life, a variety of processes are adopted. What are those? First, vacuum cooling. Second, giving gamma radiation and storing at 50 degrees centigrade. Gamma radiations are passed and the cultivated mushroom is kept at 15 degrees centigrade. After that, freeze drying in a solution of citric acid or ascorbic acid or brine, and these are the steps of this preservation. Clear? In this way, this chapter, that is importance of fungus or importance of bacteria and fungi, is completed. Okay.